clearly have to say, being at Stanford makes me feel at home. It's really something, once you've been here, you'll all have this experience. You actually feel you're part of Stanford, and when you come back, this feeling comes immediately. And yes, it is a sad part with Hardin McConnell, but I have so many good memories about Hardin that it is not only an honor for me, it's a real pleasure to give this talk. And in so many occasions, I have to think back on what I learned from him. He was a great teacher to me, and we'll see several examples on how that influenced me. And that's a little bit an overview on what we're doing as kind of research, and you'll probably recognize this one sign over there, which I stole from W.E. Murner. I borrowed it. And it's an advertisement for a Gordon Research Conference which deals with single molecule approaches to biology. And the clear message is no ensemble average. And this is basically the theme of our research in the same way as it is for W.E. Murner. And it depicts a little bit what we're doing. And many of these experiments that we do nowadays have been inspired by work of Hardin McConnell that we did discuss, because I had him afterwards. Once I was postdoc with him, I had him as an advisor. And I would always go to him when I had too many projects and I had to actually kill one of them. I asked him and he made the suggestion, yeah, cut that one. And that always turned out really good. It was really a great thing. So for now, for today, I'd like to focus on these 4 topics here and try to make the link between force and function, and particularly how biomolecules do this link. The relation with Hardin was not always easy. It could happen that he would come in the lab morning at 9 o'clock, and I could hear his voice, his dark voice, "Hermann, what have you discovered today?" And it was this today. I had barely had my first coffee, and he was expecting me to have really these results. And if he was not too happy about my answer, it could as well be that he said, "Well, Hermann, why are you wasting your and my precious time?" But as I said, we became friends. He became the scientific grandfather of my children, and both of them are in science now, so he did a good job there, and Sophie helped with it. So the topic that I want to talk today about was to a large degree actually initiated by this research I did when I was postdoc with him. And as Steve had already mentioned, we had built up supported bilayers and had put in MHC molecules, and the question was, at that time it was not clear whether a complex of the peptide and the MHC molecule was recognized by the T-cell receptor, or whether it had 2 different binding sites. And we could clearly show by combining total internal reflection excitation with first energy transfer between a donor and acceptor on these molecules that the T-cell sees a complex between them. And that was very helpful, and from that point on, the research on these MHC peptide complexes took up. Hardin McConnell was really very fond of this project, and eventually we learned that, yes, it is a very tight complex between the MHC molecule and the peptide. And then the discussion came, how can one actually quantify something like that, which has an off rate which is so exceedingly slow that it's hard to quantify? And parallel to it was actually, again, Stanford, a real big invention happening in a collaboration between Gerd Binnig and Quaid, who actually, Gerd had a sabbatical here, together with IBM Alman Labs, where Christoph Gerber had his sabbatical, and they invented the AFM. And it was just at the time when I was postdoc here, and it was when all the postdocs between IBM and Stanford, and also at that time Xerox Lab, would always meet on Saturdays and exchange information, and there it became clear this is a really hot topic. And when I went back to Germany, I actually focused on atomic force microscopy. And then again, instead of, or not only focusing on the capabilities of the AFM to image, we also looked into possibilities to use this instrument to measure forces at extremely high precision, because that's what this instrument can do too. And we learned that we can do that with piconewton precision. Coming back to that question, how can we understand the binding of a peptide in the pocket of an MHC molecule? The discussion came back to, well, can't we pull that out? Can't we really force that peptide out of the binding pocket and accelerate this process, which otherwise is extremely slow? And by means of this, learn how this process works, how this molecular recognition in the binding pocket occurs. Stanford comes into play again. Hardin had a really talented student, Vince Moy. He was a little bit chaotic, but he was brilliant. He came to my lab as a postdoc and picked up this idea that we had discussed with Hardin. Isn't it possible to pull that peptide out? Now, it turned out that in a physics lab, preparing MHC molecules didn't really work well. And so he actually looked for a system which had similar off rates, and that was at that time biotin-avidin, and that was something you could handle easily. So he started to pull on the biotin that was stuck in the binding pocket of a streptavidin or an avidin, and he could actually find discrete peaks of forces, if you look at that histogram. And what we could do is identify the binding forces of individuals. The problem was we had several of these peptide of these avidin molecules at the AFM tip, and so we had multiple bindings. We had also double binding sites here. So that was not really a well-defined system, but we could identify the binding forces of an individual, and it was soon after that by the group of Helmut Grubmüller in molecular dynamics calculation confirmed that this is the process at which this thing comes out of the pocket. Nowadays, we have a much better control, obviously. And what we do in systems like that, and that's probably the most recent version of understanding how this molecular complex comes about and the force, we take a tetramer, which consists of one active unit and 4 dead units, so we have only one binding site. This one has in addition a cysteine, so that we have a well-defined coupling of this molecule to the tip with a spacer. And in addition, we bring in a fingerprint protein, which upon pulling on this thing unfolds because the force goes through all this complex, and we designed this fingerprint protein such that it unfolds first. So this is the project that Diana Pippick and her group push in my lab, and this here gives you the tetramer of this unit of this streptactin, which was selected to bind a peptide instead of the biotin. And this is the peptide. It's 7 segments long that is recognized. This is the active unit, and those are the 3 dead units. You actually co-assemble them, and this is then the handle that you have on the molecule. So all the force goes through this molecule, and those actually are bystanders. Those are the mutations that make this molecule a dead molecule. And this is then what you get as a result. You start pulling on this complex, and in order for you to understand how these curves should be read, why don't we do it that way? So what we have is we have a protein which is folded. In this case, this is GFP. So we fold the GFP. Have polymeric anchors at which it is attached to the tip, have it at the surface attached. And if the distance is such that the polymer in between can fluctuate, it explores all degrees of freedom. There is no force acting on the tip. Now what we do is, if we separate the tip from the surface, we actually freeze out degrees of freedom. That costs entropy. And if you look at the term of the entropy in the Gibbs free energy, so that means costs Gibbs free energy, gradient of Gibbs free energy is a force, so a force builds up polymer elasticity. So if we separate it more and more, what happens is that first, this protein unfolds, the barrier is overcome. Now hidden length is added to the chain, the force drops again. This is then right here. Oops. The force drops again. There is an intermittent step here that comes from the substructure of the thing. And finally, if we pull on the thing again, then the anchor, oops. In this case, the streptavidin ruptures and the force drops to 0. So this is how you should read this kind of curves. You will see them more frequently. This is a way to determine barriers in the folding landscape of proteins or unbinding forces of ligand receptor complexes. Now, if you vary the rate at which you do that, then you'll find out that the distributions, the histogram, the maximum likely unbinding force, shifts to higher forces if you pull faster on the thing. Now, this envelope here is the unfolding of the GFP. Doesn't move that fast as the envelope of the streptactin-streptavidin interaction. How do we understand it? Well, what we have is a combination of thermal fluctuations and mechanical force acting on that ligand when we pull it out of the membrane or when we unfold a protein. So it's this interplay. It's not only mechanics and it's not only fluctuation. So if we have no force acting on it, then yes, if this is the binding energy landscape of this complex, then thermal fluctuations will kick it around and eventually the thing will unbind. We have thermal unbinding. And this is the simplest approach to do it. Obviously, one can do that much more precise, but it will always be of that kind that we have a Boltzmann factor with the energy in it kicking it out. Now, if you force the system, then you choose a direction at which the complex comes apart. It may well be that you chose a direction where there is an additional barrier. So you'll have a path-dependent additional term in this process. But what you do is basically you drag the system out of this pocket and you do mechanical work on it. And this mechanical work is integral f dx because it takes force to drag it out of the valley. If you approximate that with an approximate potential with delta x, then you can write it like that. And what it tells you is that the forced unbinding rate is the thermal unbinding rate, then comes a factor that has to do with the path. And here in the exponential is the force times delta x. What that means is if you take now the logarithm of it, then you clearly see that the force goes like the logarithm of the rate at which you do the experiment, and this is basically what we find. If you do that in this case with the streptactin and the peptide, you have the chance to grab the peptide at two different ends, either at the C terminal or at the N terminal end. Obviously, the binding energy is the same, but what we measure is when we do it on the C terminal end, or if we do it on the N terminal end, are clearly different forces and clearly different force dependencies. What that means is that the system chooses a different path to get out of the binding pocket. So we force it along a different path, and as a result, we get different potential width. In this case, this is on the order of 2.3 angstrom, and in this case, it's on the order of 4 or 5 angstroms. And those are typical numbers for such small molecules and for the unbinding of it. So what it tells us is the energy landscape is a means for the molecule to determine whether high forces or low forces are required to actually pull it apart. And if you now see that in a big picture of molecular motors, that's probably the weakest. I'm not sure, Steve, if you agree with these numbers, something like 5 piconewtons and something like 5 kT as energy. And at the covalent end, we have on the order of 1,000 piconewtons as unbinding forces and 140 kT as energy. If you look at, for example, DNA, it's made to actually keep both strands together. So the binding energy is relatively high. It doesn't cost much force to separate the two strands. That's its job. It should be read easily, but if no force is acting on it, it should stay stable. Molecules, for example, like streptavidin and streptactin, they're actually selected to hold in the selection process. So they create quite sizable unbinding forces at comparably low energies. There is one extreme case, which we'll talk more about, the Dockerin cohesin case, which has evolved and was selected to withstand forces, and there are special mechanisms for these low energies and extremely high forces that have evolved in the development of that molecule. It is part of an extracellular organelle called cellulosome. And the cellulosome grabs a cellulose fibril, and its job is to disintegrate it. And the question is now, how are forces, this is the cellulose fiber, this is the surface of a bacteria, how are forces actually propagating in that? And this project was driven by Michael Nash, Konstantin Schöler, Klara Malinovska. Those are the main players in this project. Why are we interested in that? Well, one of the reasons is that a big share of the world's biomass is locked up in lignocellulose. Lignocellulose is crystalline cellulose and wrapped with lignin, which stabilizes it. And altogether, it makes a fiber which is extremely hard to digest. And certain bacteria and fungi have developed organelles that are capable of doing it, multi-enzyme complexes. And one of the goals is obviously if one can convert waste into biofuel, then this might be a substantial share of our future energy sources. These extracellular organelles, they live at the surface of bacteria. This is a thermocellum, and those humps here, they're the organelles by which these bacteria hold on to the cellulosome fibers and digest them. Now there's one problem. If a bacterium holds on to the cellulose fiber and does it with non-covalent interactions, these interactions have to be extremely strong because these, for example, live in the stomach of a cow that digests straw, and the environment there is extremely turbulent, forces acting on it, and since it's a non-covalent interaction, it requires special tricks to allow that. The reason why we're interested in this is the following. First of all, we'd love to design these cellulosomes, which are at the surface of the bacteria, redesign them in order to use them for artificial multi-enzyme digestion of cellulose fibers. This is clearly one of the goals. It turns out this is an extremely rich toolbox for non-covalent interactions, all protein-based. Because what it is on the surface of the bacteria, there's first a cohesin molecule, and the Dockerin molecule then attaches to it, and what we have here are lots of different cohesins which are building something like a Lego plate. And the bacteria has on the order of 20 or 30 different genes for enzymes. Those are lignases, cellulases, beta-galactosidase, and so on, and plugs them onto this Lego board. So it has a certain variety of enzymes that it uses to digest the lignocellulose. And on the second branch, it has cellulose binding modules where it holds onto this fibril. And all these molecules here have to be stable because they're at the outside of the bacteria under worst conditions. They live in the cow stomach. They can also live in the soil. And this is an ideal background if you work with an antibody that's used to nicely buffer pH and so on, because it lives inside the body. These live outside a bacterium and have to survive basically everything. So if I can give an advice, these molecules are extremely nice, and some of the functions are orthogonal, some of them not, for non-covalent attachment, and you have a huge variety. And these molecules are rather small. So if you look at the Dockerin, it has 20 kilodalton. The cohesin is a little bit larger, but there are also smaller cohesins. And what we're doing at the moment is we're actually varying the species and then use all these combinations and look at the cross-reactivity. And typically, what we get is interaction forces on the order of 150 piconewton. So they're on the ballpark of what you would also get for a biotin-avidin interaction. And you have a huge variety of molecules that you can use. Now, how do we handle this variety? Because these proteins can't be produced by culturing the cells. These bacteria are typically not culturable. There are 2 or 3 strands which are standardized in labs, but we have all the genes available. So if there is a way to go from the genes right to the protein in an efficient way, this is the way to go. And so we teamed up with Steve Quake. Stanford comes into place again. Steve has developed his microplumbing, this rubber microplumbing, and we make use of it. And the idea behind it is the following. He has this microfluidic system where he can address individual reaction chambers, and there are thousands or so on a chip. And then what we do is we take one of these chips and plot the DNA of all these different proteins. And now comes the trick. This is the gene of interest, the one that we want to express. And we frame it with the YBBR tag. That's a tag which covalently attaches to coenzyme A with the help of an enzyme, and at the other end, a Dockerin. We actually have it in the DNA chamber. This is what we have. Those are the DNA spots. And we can use all the buttons and valves of this microfluidic system to cut off the reaction chambers. So in this case, we would now have here the production of the gene product. If we flush in an in vitro expression system, what happens then is that we produce the product of these genes. And if we pull back the plunger for a little bit, let the product bind to the surface is pegylated with coenzyme A, all the products that have a YBBR tag expressed will now covalently attach themselves to the surface. If we pull back the plunger a little bit so that at the rim we can decorate now the Dockerins with a cohesin that has a fluorophore, we can quantify the efficiency of the in vitro expression, and the result is actually something like a target, because around this plunger here we'll have now a ring that is decorated by the fluorescently labeled cohesins, and we now in the middle have the spot where we have the protein produced and covalently attached to the surface. And now if we do an experiment by removing the microfluidic system, we can approach it and do now a single molecule force spectroscopy, and we get only those that have both of these, the YBBR tag and the Dockerin expressed, so that are fully expressed proteins. All the ones that have not actually produced a full-length protein are not part of the experiment. This allows us now to do really large scale, because as I said, there are up to 1,000 spots that we can put on. This allows really large scale screening now, and what we have here on one chip, just for demonstration, those are fibronectin domains. This is an extracellular matrix protein. Spectrin, that's an intracellular cytoskeletal protein. Xylanase, that's an enzyme part of the cellulosome. This is GFP as a model system. We can co-express them all on one chip and have them available now for force spectroscopy. So that allows us now to screen. The big surprise came when we started to look at this guy here, whose job is to hold the bacteria onto the cellulose fiber. This is the one that takes all the load. And what we learned is that if we actually pull on a system like this, where we have now, again, fingerprint domains built into it, we learned that, yes, this is the first step where the xylanase unfolds, the next step where the cellulose binding module unfolds, and the last step where this complex here ruptures, and that's up at 500 piconewton. And we have meanwhile systems that go up to 800 piconewton. This is approaching the strength of a covalent bond. How does that work? How can that work out? Molecular dynamics calculations give an insight into it. So what we have here is this complex of the cohesin and the Dockerin, and if we apply now load and increase the force while we do molecular dynamics calculation on it, what we learn is that these fingers here hold onto the partner, and the stronger we pull on it, the closer these fingers actually hold onto it, and that can be quantified by looking at the area of contact, and the contact area is actually increasing if we put load on the system. I'm not sure whether one can call that a catch bond, but it's in any way a bond that strengthens under load. That's not necessarily a new concept. That has been used for quite some time. Stone lifters are instruments that convert the direction of a force acting on that instrument here. You pull along that line, it's converting it to hold onto these little holes in the stones. So it's a little bit like the mechanism that we've just seen. The catch bond, you have that structure, you pull on it, and it's getting tighter instead of loosening up. Well, we actually looked into other systems of the same kind and found even higher forces and asked ourselves, how is it actually if you have such a molecular complex, which at one end is attached through one covalent bond to PEG or whatever, and at the other end also it's finally one atom at which you pull. But all these structures in between, how is the force propagating through such a complex? And this is a general question for all these systems that are under force. How is force propagating in such a biomolecule? We had no clue in the beginning and just did a toy model, in molecular dynamics where we had stiff and weak springs between these atoms. Those are weak springs, those are stiff springs, and we were pulling on this guy here, and we were trying to find out how we can identify where the force goes through this very simple construct. And we looked back into the old days when people weren't capable of doing single molecule experiments and started to understand a lot about functions like this by looking at fluctuations and looking at higher moments of fluctuations. And this is actually a well-known relation, and I'm pretty sure Hardin McConnell would have known that offhand, which relates the covariance of the position of the atoms with the force propagation. And if you read this line this way around, then it says, yes, you get a displacement of the atom J by a force acting on the atom I, and the proportionality is the covariance normalized by kT. So this relation here tells us if we are capable of measuring the covariance, then we learn about the force propagation in such a system from atom AI to atom J. And if we do this for all of them, then we can reconstruct the path of the force through the molecule. Well, what you can actually pull out of the molecular dynamics calculation is the normalized covariance, and that's the Pearson correlation. And if you then plug in an approximation that you say, well, these potentials are in a good approximation harmonic potentials in which these atoms sit in the interaction with all the others. And if you then do the equipartition theorem and say, yeah, each of these degrees of freedom, it gets excited in the same way with kT, then you can write it down such that you get an expression if you know the covariance and the stiffnesses, and both of that you can pull out of the molecular dynamics calculation, then yes, you get an expression on how force is propagated in a molecule. Well, if we do this with our time model, then in red here are highlighted the pair correlations, and if you now take the one that has the strongest correlation, then in this case this is this path. So in this time model, this is the path in which the force propagates through that complex. If we apply this now to our Cohesin and Dockerins systems, and first look at the Pearson correlation. This means how is the fluctuation of atom I correlated to the fluctuation of atom J? And this is the block of the Cohesin, the first 200. This is the X module, that's the strengthening structure of the Cohesin, that's the Dockerin. Then, yes, it's clear that the correlation is strongest along the backbone. Obviously, there's nothing really surprising. But there is also off-diagonal very strong correlation. This is because of the superstructure of the folding of the Cohesin in the same way of the Dockerin. And there are also cross-correlation between the fluctuations in the Dockerin and the Cohesin that stems from the binding. This is because they bind. There is this cross-correlation between this guy and these down here. So we have an idea on the correlations. What we can do now is we have many of these correlations and have now many force propagation contributions. And it's a little bit the same problem as you have Google Maps. They have a landscape and have to give you a suggestion on how to get easiest from A to B. There's many paths which are actually going there, but they're inefficient because they're so far. Then there are short paths, and so this Google map has to decide which direction to give you, and it does that with dynamic network analysis. And we do the same thing. We take now every amino acid of this molecular complex and look at the covariance or at the Pearson correlation between these two amino acids because they're talking to each other. And now we can measure the strength of this interaction because we've developed all this algorithm. And what we find now is we have these dark lines here that represent very strong correlations, very strong force propagation, and weak ones. And if we now map this again onto the structure of the protein, we get this here. So we have one main path of the force, which is highlighted by this thick brown thing. And then we have many weak paths, which are there, but that do not necessarily contribute a lot to the force propagation in this complex. Now, the surprising thing is, if you look now at the path of the force, this is the interface between the Cohesin and the Dockerin. This is the binding interface. And what you'll see is the path actually makes a turn right in the binding interface, this hook here. So if you pull on the molecule, the force gets propagated perpendicular. And what it means is you pull on the molecule, but the force is not acting along that direction where these two molecules would come apart. It's acting in the plane. So it does not contribute to separation of the molecules. So you apply force to it, but the force goes in the wrong direction. So that's, in this case, the explanation why this molecule at a decent binding energy can withstand these enormous forces. The force is propagated perpendicular to the separation direction in the binding plane. And that's quite an interesting result, that Mother Nature finds these kind of tricks to optimize molecules for the task of holding. Let's look into a leg and look into another function that Mother Nature has optimized in terms of force. And if we look into the leg, we find mechanosensitive enzymes that regulate the sarcomer. And this is work that was done, pioneered by Elias Buchner and Diana Pippick. And we had a very strong collaboration with Frauke Gretter, Matthias Gottel, and Helmut Grubmiller. If you look into the muscle, zoom in, then this is the sarcomer. And we all know that here are the muscle motors. And I'm not talking about the motors. Steve knows much more about that. And if you have questions, ask him. But the thick filament has in the middle actually one molecule, which is the titin. And it has two functions. It first of all organizes the structure in the thick filament, but it also has mechanical function. And the key is, if you look into this filament here, there is also a kinase. And the reason for that is the following. Imagine that this one half of the sarcomer is somehow damaged. Then these molecular motors here pull, but there is nothing that pulls against it, and it will actually just pull the sarcomer apart, and the motor machines won't work anymore. In order to prevent that, titin is one of the mechanisms, but it also needs to report that some imbalance had occurred, so it needs a kinase. And what was known is that this kinase here is also talking to repair proteins and so on, activates repair mechanisms. And the way it is built into this molecule is that the force goes through this kinase, and it has two structures, this red loop and this yellow one here, that cover the active site. So it's an auto-inhibited ATPase, and the working hypothesis was this is a force-activated enzyme. Now what one can do is one can take this molecule with its adjacent proteins so that the structure feels happy, and then what one can do is successively unfold such a structure. And what you'll read is here are the barriers that are overcome to unfold Ig domains, and that front part here, that's the titin kinase. If we first look at these big signals here and just get an idea about the energetics of it, then what we have, if we integrate the mechanical work that we put into the system here, then those are on the order of 1700 kT. Compare that with the covalent bond, this is only on the order of 100 or 200 kT. And so what it means is that this here is an ideal safety belt for the muscle. And the mechanism behind it is, if you look at the structure, there is a series of hydrogen bonds which needs to be overcome and broken, and once you did that, the whole structure falls apart. So this is the main barrier which prevents the protein from unfolding and which you have to overcome by force. If you compare that to a safety belt, then it has really big advantages. This is the unfolding of a full-length titin, and if we relax it and let it sit for a while at an expanded state, it will refold. And if you actually relax it completely, it will nearly completely refold. You can, by means of this, restore the function of it. So it has a giant compliance by the unfolding of the protein. So all the mechanical work that you put into the system goes basically into entropy, hydration, and that will then actually be just translated to heat, and you can completely re-anneal it, self-healing. Do this with your car, that won't really work well. Now, if we look into the front part, where the titin kinase is, then what we find is we find a series of barriers which need to be overcome if we stretch this kinase. So we have these two front parts identified as potential candidates for these two structures that I mentioned in the beginning that cover the active site. So the idea is now, if we add ATP, then we should see whether these two are the ones that block the ATP binding site, and this can actually be done. If we add ATP, we find a third barrier, and we find this only once these two barriers have been overcome. So these structures had opened up the active site. And only after both of them have been overcome, we can really measure ATP binding. Now the question is, those are two doors. And are these two doors that cover the active site working independently of each other? So is it that if we pull on the system, this one can open or this one can open, and so they can do that statistically? Or is it that door 1 has to be opened in order for door 2 to be opened? So do they condition each other? And that's a quite interesting difference. And that can be investigated now, in this case, by keeping the force constant and looking at the elongation. So we switch basically from a Gibbs boundary condition to a Helmholtz boundary condition. We keep now the force constant, and what we get is incremental increase in the length. And if we do so, then we can measure the kinetics. This is the time already in logarithmic scale. We can determine the kinetics by which these doors here open up the active site. And if those are two independent doors, what we'll get is just an exponential relaxation. But if these doors are conditioning each other, then this means first door 1 has to be opened. And if door 1 is open, door 2 can be opened, which comes to a curve shape like that. So this mechanism here is the one that we find in the experiment, which means, yes, these doors are conditioning each other, which means this is a very efficient protection against thermal fluctuations. You do not want to have thermal fluctuations to kicking the doors open. And so if the probability is the product of individual door openings when they're conditioning each other, then this is a very efficient way to protect against thermal fluctuations. Now if we take this picture complete and compare it with molecular dynamics calculation, then those are the barriers that we identify in the AFM experiment, and this here are the barriers in the molecular dynamics simulation. This is actually the opening of the doors, and what you see here, we pull away the first part, we pull away the second part, the active site becomes accessible, ATP can bind, phosphorylation of the binding site can occur, and then molecule is activated. The student that did this work one evening went to the bar and found a student who's in the visual arts department. And they decided to make a movie about his PhD thesis. And it became a little bit pathetic, but what you'll see here is now we zoom into the muscle and see individual muscle fibers. And if we zoom into these fibrils, then we see these individual machines here, not individual machines, these sarcomers. And if we zoom into that, we see fibrils of thick filaments and thin filaments. And let's assume that one of the strands of the thick filaments got damaged somehow and is not working properly. Then what happens is that the thick filaments at the other end, they start pulling on it. And as a result, force goes through the Titin molecule instead of through the cross bridges. And when the force goes through the Titin molecule is stretched, and the kinase, which sits in the anchor of the thick filament, or in this case the Titin, in the Z plane. And if we zoom in, then we see the Titin kinase. This is this guy here. And if we pull on it, then we open the two doors, door 1, door 2. Active site becomes accessible, ATP binds, autophosphorylation occurs. And from there on, we don't really know much. This is actually in the mist. We don't really know much about the signaling, which then occurs from here on. But the physics part is pretty well captured by this movie. And so, although it's a little bit, as I said, pathetic, but that's fine. So much about what Mother Nature does in order to utilize tricks relating force and energy. There's also a way that we can make use of all these wonderful properties of biomolecules for our purposes. And in this project, Philipp Severin and Kati Falter, Uta Winken, they explored the possibility to use DNA as a force sensor. And if you think of how can you measure most sensitive, then you will always learn a null experiment is the most sensitive way of measuring something. You have a balance, and the balance you put on your thing to measure. What you do is you balance it with a weight that you know until you're at zero again. And zero compensation is still the most sensitive way of doing it. That's one thing. The second thing is, if you look at where you pick up noise, then the fluctuation dissipation theorem tells you whenever you have dissipation, you'll also have fluctuation. They're proportional to each other. Now, if you look at an AFM cantilever, then the AFM cantilever, if it's large, dissipates a lot as viscous drag, so it also fluctuates a lot. Smaller cantilevers tend to have much less noise because they dissipate less, so they fluctuate less. If you think along that line, then you should have the smallest sensor possible. So why not right away use a molecule as a sensor? And so those two things together made us believe that taking DNA as a sensor might be a useful thing. Another thing that you learn is, if you do an experiment, do it as symmetric as possible and use the break of symmetry as your measuring signal. And so one can make it the following. Let's assume we want to, in this experiment, measure a transcription factor binding to a DNA. So what we can do is we can build up a system consisting of a ddNA strand covalently attached to an LDNA strand, so those are duplexes. And in the middle we have a fluorophore. And each end of the L and the D duplex is attached with a polymeric anchor to a surface. This is an elastomer surface, and this elastomer stamp allows us now to separate the two surfaces. So we have a completely symmetric thing. This turns this way around, this turns that way around, and if we now add a transcription factor, it recognizes only the ddNA. It will not interact with the LDNA. So if it binds to the ddNA, what we do now is we separate the two surfaces. That's all we do. If we do so, force builds up in this complex because the polymeric anchor is stretched. Eventually, the force exceeds the holding force of that DNA duplex, and it will rupture. And the fluorophore will tell us if the complex was pulled to the one or to the other side. If this stabilized this interaction, the fluorophore will be pulled to this side. The other way, it will be pulled to the other side. So the only thing we have to do is now find out where the fluorophore is, measure the fluorescence on both sides, and we have basically done the experiment. Again, we do this with Stephen Quake's microfluidics chip that has this beautiful plunger. And what we do is now we put a DNA duplex, attach it covalently to the plunger, and have the other partner, a biotin, binding to the surface of a neutravidin. Once we lower the stamp, so we measure the number of fluorophores that we have on this side, red one, green one, with the fluorescence microscope, lower the plunger, allow this contact to happen, and separate it again, and just count how many fluorophores do we have here, how many fluorophores do we have here. We have two labels, one that tells us whether this contact had taken place, and the other label tells us where this bond ruptured and where then eventually the product is. So we can do that just by measuring the fluorescence, very simple. Now, if we come back to this idea of the balances. So what we do is we prepare now balances which are pre-tuned. The normal balance is completely nicely balanced. And now we build one that has a weight already here, a weight already here, a weight already here. So we build a set of balances. And the way we do it is we take a duplex that is 20 base pairs on one side, and on the other side, we take duplexes of various length. When it's also 20, then the probability for the fluorophore to end up on one side is 50%. It's a completely symmetric system. If we shorten the other duplex, then we end up at a lower. If we lengthen the other duplex, we end up at a higher probability for the other side. So this is the way we calibrate the balance. What we then do is we throw in a molecule that interacts with the DNA. In this case, this is a peptide. We did that together with Peter Derwin and Caltech, and that peptide is a major groove binder, and it stabilizes this bond. What happens is actually we get this curve here. This means the balances that we had pre-tuned are now actually shifted, and it is this balance which is now completely balanced again, which has 9 base pairs more on the other arm. So this means this interaction is the equivalent of 9 base pairs on a duplex. So that's a measure for this interaction here. We can do the same thing with eco R1. In the absence of magnesium, it doesn't cut, it just binds. It's much stronger. So it's the equivalent of 27 base pairs, what it has as a binding strength. Or if you take the tumor suppressor factor, that is something we can't even measure with this kind of balances. Now, talking to biochemists is often helpful to have these numbers in terms of base pairs, because this is something where they have a feeling for. If you want to go back to physics, you have to calibrate it. You just measure what is the rupture force of these duplexes in terms of piconewton, and then you can give the numbers in terms of piconewton. Now that we've learned that we can use DNA as a tool to measure forces and as a tool to arrange molecules, we can combine again what we've all learned with the AFM, that we have the capability of addressing a point with angstrom precision. Now that we've also developed a technique to make protein chips, we can make large libraries of proteins where we know on position A we have protein A, position B we have protein B. And what we do now is we anchor them with DNA oligos or with some other kind of non-covalent interaction, and have these molecules stored in a reversible way at given positions. We also extend the DNA with an oligo of a given length and come in with an AFM cantilever that hybridizes to this oligo. Now comes the trick. If we pull with the AFM cantilever, we have here force going through a DNA oligo where the force goes in shear geometry to all the base pairs. What that means is we have to overcome the binding energy by working F dx, by putting in mechanical work, which means if we actually shift these two oligo halves by a base pair, there is no overlap anymore. This means it falls apart. So all the binding energy is done on a very short distance, which means it's a high force as a result. Now the anchor here, however, is attached such that if we pull on it, we unzip the anchor. This means all the binding energy has to be overcome over all the length of the DNA. That means low force. What that again means is if we pull on the molecule through this handle, this guy here will open. Now we can transfer it to the construction side, where we have the same DNA, it's just attached at the 3' instead at the 5' end. If we come in, it hybridizes again in shear geometry. And this one is a little bit longer than this guy, so if we pull, as we've just learned, this guy here will open up, and we have this oligo as a handle hanging again on the AFM tip, can go over, pick up the next protein, bring it over, and by means of this construct piece by piece by piece a structure from proteins that we had stored here. That is a technology that allows us to make use of the fact that we have on the micron length scale the capability of making patterns down to nanometer ranges now and build molecular machines. And that's the ultimate goal, because we're interested in this, the cellulosome business, because we would love to build our own artificial cellulosomes. And what you see here is, this is model and those are now data. If we come in with an AFM and deposit a fluorophore here, then we can determine the forces that are required to break these bonds as a protocol that we, yes, have deposited one molecule, and the same thing we do when we pick it up. So we know during the experiment that we have, in fact, only one. If we have several, then we bring them to the dump and place them there. And what you see here is in total internal reflection microscopy, the fluorophore that we had deposited, it dies after a while, and then you can ask, what is the precision at which we can do so. If you integrate now this movie that you've just seen, then you can determine the position of this fluorophore with an accuracy on the order of something better like 2 nanometer. As long if the fluorophore lives long enough, then this is standard, what all the high-resolution microscopies can do. It's a little bit more tricky when you ask, how precise can you deposit a molecule? How precise can you paste? Because what we have is a polymeric anchor, and the reactants, they dangle on the polymer and search their partner. And so it's the diffusion range of this oligo at the tip which determines basically the precision at which we can position. That guy here is with angstrom precision, but the deposited molecules are on the order of 10 nanometer. If we have 100 nanometer pegs as spacers, that can be improved if one wishes. We may also ask, is this a robust technology? And yes, it is. Students start inviting each other for drinks or birthday cakes, and this was for a 65th birthday of Christoph Bräuchler. So it can't be too hard, and it's pretty reliable. You can really write structures with this. And if something like 10 nanometer is the accuracy that you want, this is what we have at the moment. We're shooting for something like 2 or 3 nanometers, and I'm pretty sure we'll have that soon, so that we can build up molecular machines. So coming back to Hardin's question. It was not today, it took really a few days. Altogether, yes, but I think what we've discovered is we understand a lot about the relation between energy and forces, and we understand what Mother Nature uses as tricks to really optimize the one or the other. We understand that the force propagation in a molecule may be extremely tricky and can be used to make extremely strong bonds in terms of forces at the expense of very low energies. And Cohesin and Dockerins systems have really optimized that. Titin has a giant ductility, and it does so by reversible unfolding of these domains. They don't rupture, it's hydrogen bonds that rupture, and it's all converted to heat, but it's healing again. And I wouldn't know any material that has comparable properties. For the Titin kinase, we learned those are 2 doors that block the active site, and really it has to be a double-hit action, and that suppresses thermal fluctuations. We learned that transcription factors hold very tightly onto their DNA, and we learned that single molecule cut and paste is a robust assembly technology. Let me end by thanking the team that did it all. Here we're on the way to the Oktoberfest, and we don't look every day like that. So it's and actually it's also a reference to Hardin McConnell that I wear this Bavarian jacket. I did that from time to time when I was in the lab, and he appreciated it a lot. So I felt I should do it. Michael Nash and Diana Pippick, they're driving forces in these projects. I told you already, Elias Buchner left already, Markus Ott and Philipp Severin. Those guys did a lot on the Cellulosome system. Markus Jobst and Wolfgang Ott built a lot of the machinery. And we had very strong collaboration with Helmut Grubmüller, doing molecular dynamics calculation. Also with Zahn and Klaus Schulten, again, molecular dynamics. Matthias Gottel was the Titin kinase. Ed Beyer, whenever it comes to cellulosomes, he's the guru. Steve Quake helped us with the microfluidics, and Philipp Tinnefeld and Frauke Gretter were helpful discussants. And we're at the moment 2 postdoc positions open. If you want to get used to one of these dresses, feel free. Thanks for your attention.